Hello, good evening. My name is Hunter, and this is Content Review for Principles of Biology. Today, we're doing a little bit more of the nervous system and getting into muscles. I really like this worksheet because it's taking some of these new concepts that we talked about last week and putting it into a context that is very experiential and giving us a little bit more focus, I think, that makes it a little more understandable. I've given myself a little head start here with some drawings that we will come back to in a minute. Um, Yeah, this first question is a good review one, so we'll jump right in. So in the following questions, test your understanding of the various parts of the nervous system by asking yourself what would happen if some part was damaged. Pathology is always a really good way to test your understanding of what normal function looks like. What would the system still be able to do? What would it be unable to do? So in this first one, some nerve gases and insect poisons work by destroying acetylcholine esterase. Acetylcholine esterase is normally present in acetylcholine synapses and acts to degrade acetylcholine. What is likely to happen to nervous transmission in insects exposed to this type of insect poison? So, in the central nervous system of most vertebrates, here's our review part, right? An action potential travels down our nerve cell. It reaches the axon terminal here in the synaptic cleft. Last week we were talking about nerve cell to nerve cell junctions. Today we're talking about nerve cell to muscle junctions. And that action potential travels down the axon in the form of this influx of sodium, which depolarizes the membrane. Once it reaches the end here, that triggers an influx of calcium, which triggers the exocytosis and release of acetylcholine molecules into that synaptic cleft. And acetylcholine binds very nicely to these cholinergic nicotinic receptors on the next nerve cell or muscle cell. So acetylcholine is a very important molecule, and it will just stay there forever unless it is broke down by acetylcholine esterase. And esterase uh, gives you some indication of its function. Esters you will learn a lot about in organic chemistry, but ase um, is a very strong indication that this is a enzyme. That is important vocabulary to have in the back of your mind. So it breaks down acetylcholine so that it can be parts of it will get recycled. So acetylcholine in most vertebrates, this includes bugs. Well, no, it doesn't. In the central nervous system of vertebrates, acetylcholine can cause inhibition or excitation of the postsynaptic neuron. Excitation is we're going to keep that action potential going. Inhibition is going to be we're going to calm that down, right? So which of these it does depends on the type of postsynaptic receptor present. If so, in neuromuscular junctions, like the one I have drawn here, that is voluntary muscle, acetylcholine is excitatory, and that is true for very many of the uh, cholinergic receptors in our body. So in heart muscles, though, the effect is inhibitory. The effect of acetylcholine is to reduce the rate and strength of heart muscle contractions. So if acetylcholine esterase is destroyed, any acetylcholine released into a synapse will remain there. We kind of talked about that already. And as a result, the postsynaptic neuron will be continuously stimulated by acetylcholine to either depolarize in excitatory neurons or um, hyperpolarize if it's inhibitory. Um, so if no enzyme is available to remove acetylcholine, then it will stay. In the synapse and continue its effect. So this could be a huge problem, right? If we're talking about skeletal muscle and it keeps sending excitatory signals, it will just contract and never let go, right? And this can be especially problematic if we're talking about cardiac muscle, like your diaphragm, right? You can suffocate because your diaphragm can't do its job. On the other hand, if it's cardiac muscle and acetylcholine usually has that inhibitory effect, it can depress your heart rate really tremendously. So that combination can be lethal pretty quickly. The pufferfish, or fugu, contains the poison tetrodotoxin. Some shellfish produce a paralytic poison called saxitoxin. Both of these poisons block the sodium channels in neurons. What specific effects could these toxins have in neuron function? Let's go back to this drawing I did, right? So sodium channels are responsible for that action potential traveling down the axon, right? And if those sodium channels can't do their job, sodium stays outside of the axon, then there's no depolarization, right? So if the sodium channels are blocked, axons would be unable to generate or propagate action potentials. Sorry, I'm making myself dizzy. Axons unable to generate action potentials. So if there's absolutely no action potential going on, it depends on the neuron. I was about to say that this might result in flaccid paralysis, but we also have neurons that are inhibitory, like we were talking about. So depending on which ones are affected, you could have flaccid or spastic paralysis. A type of spider, the funnel web spider, produces a toxin that blocks calcium channels. Can a neuron exposed to this toxin fire an action potential? Can it fire an action potential and explain? So this one's a little bit different. It's not just how will it affect the neuron. It's asking specifically about action potentials. And because sodium, like we were talking about before, sodium channels are the ones really responsible for the propagation of that action potential. They should still be able to do it, but this is also going to affect other neurotransmitter signaling in that synapse, right? Because calcium is the trigger for releasing a lot of those. So neuron can still fire an action potential, probably, because these are sodium driven, but other signaling maybe may be impacted. When we get into neuromuscular junctions, you're going to see how this will absolutely have an effect. Can a neuron transmit a signal across the synapse using neurotransmitters? That was part two of the question. Um, in chemical synapses, the influx of calcium is required to cause the synaptic vesicles to fuse with the membrane to excitose from the presynaptic neuron. So if calcium channels are blocked, no release of neurotransmitter from the presynaptic neuron. Therefore, signal cannot be transmitted across the synapse. And again, just to visualize that, the action potential arrives at the end of the axon. We get that influx of calcium. If those are blocked, the acetylcholine is going to stay right where it is, or other neurotransmitter. All right. You isolate a section of Squid giant axon like we were talking about last week, and arrange an experiment so that you can change the solution bathing the axon. You insert an electrode into the axon and place another electrode outside the cell so that you can measure the potential across the cell membrane. Let's draw this. We have our axon, we have our voltmeter, and we have a probe in the solution and a probe in the cell. With the axon bathed in normal extracellular fluid, you observe a resting potential of -70 millivolts. And when stimulated, positive 55 millivolts. So that's that depolarization, right? We're given some values. You change the solution bathing the neuron by increasing the potassium concentration to 40 millimolar. What effect will this have on the neuron? For example, will it depolarize the membrane and make it easier to start an action potential? Or will it hyperpolarize the membrane and make it more resistant to starting an action potential? Or will it have no effect? And explain your answer. Now, we could go back and find that formula from the previous worksheet and actually do this quantitatively, but the important thing is that we understand this conceptually. If we're looking at the inside of our nerve cell, potassium is what's high, and sodium is what's high on the outside. It's that salty banana, remember? So if we add potassium outside of the cell, is this going to have is it going to increase the difference on either side of that cell membrane, or is it going to decrease the difference of potassium on either side of that cell membrane? The answer, of course, is that if we're adding potassium to the outside, where it usually wouldn't be, then it's going to even out the difference on either side, and that is going to depolarize. And I'm sorry, I'm reading the wrong question. The answer is going to be, yep, yeah. Also important to remember that the charge is generally negative inside the cell. And I just want to make sure I hit all of my points here. Yeah. So adding potassium outside the cell or in the fluid will depolarize the cell membrane. And this is either going to have the effect of permanently lowering the threshold so that action potentials fire more easily, or they'll just fire constantly. And that is something that we would need to quantitatively calculate. What would happen if instead of adding more potassium to the outside, you added more sodium to the fluid bathing the neuron? So same question. Normally we have, excuse me, normally we have high potassium inside and high sodium outside. What would happen if we added even more sodium? This would increase that difference even more, right? So hyper, it's going to change that. Yeah, I want to make sure that I'm emphasizing this appropriately. When you decrease the difference, the electrochemical gradient across the membrane, you are depolarizing, you are reducing the difference in voltage. And if you increase the difference, then you are hyperpolarizing. And hyperpolarizing, we know, is an inhibitory signal. Yeah. As in the first part, sodium ions cannot be added without adding a counterbalancing anion, for example, chloride, without having some effect. The addition disrupts the sodium and chloride equilibrium potentials, and as before, the axon is more permeable to chloride. So we could even out all of these changes by adding chloride ions, and that would mellow this out. But permeability of the cell to chloride is very different from sodium or potassium. As before, the axon is more permeable to chloride than to sodium, so chloride ions will move into and hyperpolarize the axon. In both of these questions, the sodium-potassium pumps that we talked about last week will eventually restore that balance. Cool. Any questions so far while I get ready for the next one? Making good time today. All right. How is the nervous system organized? Neurons are single cells composed of processes called dendrites that carry information to the cell body, and an axon that carries information away from the cell body. In simple terms, nerves are bundles of dendrites or axons, or mixed axons and dendrites. Ganglia are collections of cell bodies, and brains are collections of ganglia. Processing and integration of the information occurs as a result of interaction within and between ganglia. So responding to a stimuli requires, at a minimum, reception of a stimulus, which triggers an action potential in a sensory neuron, which synapses with the central nervous system and generates an action potential in a motor neuron, which causes a response in a muscle or gland. And we have this nice visual here. So we start with a stimulus, sends a signal up the axon to the central nervous system. Here it's synapsing with the spinal cord. This would be a simple reflex arc because we're bypassing the brain, although the brain could be included here as well. And then it sends an efferent. Yes, we're looking at a motor neuron here, so this would be an efferent signal back down the axon to some effector. And in this case, it's skeletal muscle. Cool. We're asked to draw it here, but they've already done that. The blink of an eye response is one type of simple reflex response that occurs, for example, in response to water splashing up out of the sink or to a snowball aimed at your face. When this occurs, you blink, and only afterwards do you realize why you blinked. What would you have to add to your diagram above to allow you to realize why you blinked? So the nice thing about those reflex arcs is they bypass the brain, right? They are hardwired. They are extremely difficult to overcome. We would have to add some kind of additional brain circuitry. So you need to add an interneuron or another synapse to the sensory side of the axon that led to an interneuron, which would carry the information to the brain to allow you to realize what happened. Needs a neuron or interneuron from the afferent, that is sensory neuron, to the brain. And that would allow you to consciously be aware of what happened. So why does it take you longer to realize why you blinked than it takes for the blink reaction to occur? Again, these simple reflexes are hardwired. They are very short, very fast circuits. The simple reflex, the sensory to motor neuron, cuts out the middleman, cuts out the brain, right? So, simple reflex bypasses the brain. which allows reception and interpretation functions of the brain to occur normally when you have that going straight to the brain. So this means that the protective response, the blink, can occur as fast as that action potential can fire. Most such reflexes are designed to protect or ensure the livelihood of the organism. What composes the central nervous system, or the CNS, and how does it differ in general form and function from the peripheral nervous system, the PNS? This is my very cartoonish brain and spinal cord. Actually, I'm going to move that over. The CNS is the integration and processing center. I'm going to keep doing this in blue, actually. Integration and processing. As such, it contains large collections of cell bodies and interneurons, lots of cell bodies and interneurons. The orange. This is CNS, brain, spinal cord. PNS. The peripheral nervous system contains sensory neurons. So we're going to go, it's fine. We're going to go this way. Sensory neurons, which carry information from the sensors. We'll do a little box here as a sensor. To the central nervous system, but also to motor neurons, which carry information from the central nervous system to the effector. So flow of information is in that direction. And that's about as detailed as we need to get. Yeah, central nervous system is the brain and spinal cord. The peripheral nervous system is everything else. The autonomic nervous system is subdivided into the sympathetic and sympathetic and parasympathetic systems. How do the general function of each of these divisions interact to maintain homeostasis? Sympathetic, the most important thing that you can remember about this one is it is the fight or flight division of the autonomic nervous system. So autonomic is the umbrella canopy. This is all stuff that is automatic, things that are not under conscious control, okay? So autonomic, not under conscious control. Sympathetic is fight or flight. This amps everything up, right? Gets you ready to exercise, to run, to fight that bear, right? Responses by the sympathetic nervous system are often called fight or flight responses. These tend to upregulate conversion of glycogen to glucose. So glycogen is the storage form of sugar to glucose in the bloodstream, the energy that we can use to turn into ATP for activity. They cause the secretion of epinephrine, which additionally upregulates a lot of these processes and increases heart rate, blood pressure, flow of blood to skeletal muscles and the brain. So whoa, buddy, come on. So we have a general increase in blood glucose, heart rate, blood pressure, blood flow to skeletal muscle. We have increase of epinephrine or norepinephrine. Increased blood flow to skeletal muscle and the brain. So sympathetic, ramping things up. Parasympathetic is winding everything down, down regulation. This is your rest and digest division. The parasympathetic system acts antagonistically. It has the exact opposite effect to the sympathetic division. Antagonist, anti, against. You may see these abbreviated, the PNS and the SNS, although it's less common. Parasympathetic, rest and digest. When stimuli to the sympathetic system are removed, you remove the stress, you remove the sense of the danger. The parasympathetic system promotes calming activities. So reduction in heart rate and blood pressure. You'll get increased digestion. And increased glycogen production. So glycogen again, we're taking glucose out of the bloodstream and putting it as glycogen in the liver. Decreased blood glucose. One exception to this is control of reproductive organs, where the sympathetic and parasympathetic systems actually complement each other or they work in concert. All right. Fibromyalgia is a condition that affects about 2% of the US population. You may have seen some commercials about it. Its primary symptoms are chronic pain, difficulty sleeping, and fatigue that is not relieved following sleep. Other reported symptoms include jaw clenching at night, dryness of mouth and eyes, an increased need to urinate, irritable bowels, muscle numbness and tingling, and headaches. Several researchers have proposed that fibromyalgia results from failed regulation of part of the autonomic nervous system. If this is true, failure of the regulation of which division of the ANS could account for these symptoms, and explain your reasoning. So looking at these symptoms, clenching of the jaw, sounds like we're a little stressed out. Dryness of the mouth. We have, this is not testable, you don't need to remember this part, but we have salivary glands that produce serous and mucosal secretions. Mucosal is the very sticky stuff. Serous is very watery. The serous ones are stimulated by parasympathetic activity, and the sticky, the mucosal ones, are stimulated by sympathetic activity. So this is called dual innervation by competing pathways. Again, not testable, but it can kind of help you narrow it down, because drooling a lot is not necessarily a symptom of one or the other, it's what kind. Other telltale signs: increased need to urinate, irritable bowels, muscle numbness and tingling, and headaches. So looking at all of these, it seems like these are really generalized symptoms. But given these symptoms alone, you could propose that the illness is caused by hyperactivity of which system? Given symptoms could be caused by hyper, that is overactive, I'm going to say PNS. So if the parasympathetic, the rest and digest part, is overactive, you're going to get this feeling of needing to rest even though you've slept. You're going to have to go to the bathroom even when you just went to the bathroom. You're going to have, yeah. I threw you guys a little bit of a red herring with the dual innervation of the salivary glands because that's just confusing. Um, but like if you've ever, a lot of endurance athletes, runners, soccer players, will deal with cotton mouth at some point. Or you've been running, you've been sweating a lot, and your mouth feels really, really dry and it's just very sticky. That's in part what's going on there. Um, so hyperactivity of the PNS could account for all of the symptoms with the exception of the dry mouth, muscle problems, and headaches. But the digestive problems are a really big giveaway here. So to test their ideas, the researchers recorded the heart rates of both controls and individuals with fibromyalgia for 24-hour periods of normal activity. They observed that patients with fibromyalgia showed greatly elevated heart rates both while active and when asleep. Does the addition of this information change or support the hypothesis you developed in the previous question? So heart rate remains elevated. If sympathetic increases heart rate and parasympathetic decreases heart rate, can we attribute this to parasympathetic overactivity, right? If it's parasympathetic overactivity, wouldn't that bring the heart rate down? The primary symptoms appear to be the result of hyperactivity of these primary symptoms appear to be hyperactivity of the sympathetic system. Of the sympathetic nervous system. I may have said parasympathetic. I'm sorry. Sympathetic system. Hyperactivity of the sympathetic system, the SNS, could elevate the heart rate and blood pressure and lead to difficulty sleeping and chronic fatigue. Prolonged exposure to high levels of adrenaline associated with hyperactivity of the sympathetic system also leads to a reduced stress response. This is a desensitization to stress, basically. So if chronic, you get a decreased stress response, which may then start to look like parasympathetic overactivity because your sympathetic is no longer doing its job, right? So researchers propose that this may be the cause of the irritable bowel and the bladder issues, as well as some of the limb numbness and the dryness of the eyes and the mouth. Is your yeah, yeah, you're so amped up for so long that the stress response stops being able to do its job. Well, how does sarcomere structure affect muscle function? Looking at our time, we're doing okay. All right. So switching gears a little bit, we're going to the muscle side of the neuromuscular junction. Sarcomeres are the unit of muscle contraction. Sarcomeres are arranged linearly in myofibrils, myo meaning muscle, fibrils being fibers. Excuse me. Myofibrils are arranged linearly in muscle fibers. Muscle fibers are organized into muscles. I'm going to draw this a little bit differently than I was planning on. So you have, well, yeah, we'll go left to right. So we have our muscle. And that is divided into muscle fibers that are all oriented in the same direction. And within that muscle fiber, we have myofibrils, which are all oriented in the same direction. Within a given muscle, the muscle fibers all oriented in the same direction. You can find some really good illustrations of this online, but I may actually have a good one right here. Yeah, here we go. Let me see if I can just copy and paste this in. Ta-da! Okay, so you have your skeletal muscle. We have a muscle fascicle, is a unit of muscle, which is divided into individual muscle fibers, and then the muscle fiber is divided into myofibrils. So a little bit clearer than my cartoon illustration here. And a sarcomere is that single unit of contractile ability where you have actin cycling on myosin. And as the muscle contracts, those actin filaments move towards the center of the sarcomere. I'm just getting into our sliding filament model. I'm getting a little ahead of myself, but this is good preamble. Come on. Describe the sliding filament model of muscle action. For example, what interactions power the movement of filaments in association with each other? Include the troponin complex, calcium binding sites, tropomyosin, myosin binding sites, and actin in your description. Okay. So as drawn above, you have actin on myosin. Myosin are your thick filaments, and myosin has these myosin heads kind of staggered on it. Myosin wants to bind to actin. The problem is actin is protected by tropomyosin. I'm gonna actually get my notes in front of me so I don't fly off the rails here. Yeah. Cool. So black fibers are actin, orange is tropomyosin, and the blue is myosin. Now, actin has these binding sites on it, which want to bind with myosin. So these are your myosin binding sites on actin. As long as tropomyosin is in place, the myosin heads cannot bind with those myosin binding sites on actin. The way that we get rid of tropomyosin is with an influx of calcium, and calcium is going to bind to the calcium binding sites on tropomyosin. So this first frame here is at rest. No binding is happening. When we get an influx of calcium into the sarcoplasm, sarcoplasm is the area around these fibrils, right? Just recreating our actin and myosin here. With calcium bound to it, tropomyosin pulls away. And it uncovers the myosin binding sites on the actin so that the myosin heads can grab on and, like hands pulling hand over hand on a rope, can pull the actin along the length of the myosin. So calcium is present. Myosin head binds to actin and pulls it. And muscle contraction occurs. Make sure you can see all that. Cool. So we've got the troponin complex, we've got calcium binding sites, myosin binding site, the actin. And so that's kind of all the moving bits and pieces except for ATP. And ATP is the trigger for the myosin heads to stretch like a rubber band and to pull when they dephosphorylate, expending that energy in that high energy phosphate bond and pull the actin along. So this is the sliding filament theory. The myosin remains pretty much stationary and it pulls the actin along it to contract those muscles. So how do your answers to questions 1 and 2 help explain why at maximum contraction a muscle measures 70% of its extended or uncontracted length? So it was at 100%, and at maximum contraction, it shrinks down to 70%. So during contraction, the actin filaments are pulled towards the center of the sarcomere by those myosin heads, like we talked about. I want to make sure that this illustration is still visible for you guys. I'm going to do green so the actin is sliding that direction. And you would have myosin heads oriented in the other direction on the other end that are pulling actin towards the center. We'll call this the center of the sarcomere. So during contraction, the actin filaments are pulled toward the center of the sarcomere by myosin heads on the thick filament. At maximum contraction, the sarcomere width is equal to the length of the thick filaments. In the relaxed sarcomere, the length of the thick filament is about 70% of the sarcomere width from Z line to Z line. And Z lines, yeah, I can draw this. Myosin, actin, 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 actin. There's our sarcomere. And then we've got the next myosin and the next actin. And in reality, you would have these kind of stacked. The Z disc is this stacked area. And it actually looks like a Z, a bunch of stacked Zs on the microscope. So that's your Z disc. When we have a contraction, actin pulls towards the center. So the Z discs move closer together. So thick filament is 70% the length of the relaxed sarcomere. This is kind of a good opportunity to talk about overlap too, because the way that I've illustrated this kind of doesn't show the difference in length. So the thick filaments are shorter. And I totally lost my train of thought. That happens. We'll see if it comes back to me. At maximum contraction, the sarcomere width is equal to the length of the thick filament. And the thick filament is 70% of the relaxed sarcomere length. You can check this by measuring the width of the relaxed sarcomere and the contracted sarcomere in your textbook. Review figure 45.6 and the associated text in biological sciences. Then answer these questions. Acetylcholine in an excitatory neurotransmitter is the neurotransmitter released into the neuromuscular junction. This should be a review for us at this point. Ooh, I see we have a question in the chat. This is very important. So ATP is really, really important in this whole sliding filament model, and it's worth spending a little bit more time with, actually. So there's our actin and myosin again, and here is our myosin head in its relaxed, chilling out state. And again, I'm going to pull up my notes here so that I don't fly off the rails. Show me binding with ATP, please. Pretty please. There's the structure. Oh no. I guess I'm just going to have to do it from memory. Okay, so I want to zoom this in even further before I talk about it too much more. The myosin heads are actually made up of two pieces that are kind of braided together. One of them, we'll call it the orange one, is the one that binds to actin. That's the actin binding site. And this blue one is the ATP binding site. Make sure you can see this. When an ATP comes along and binds to the myosin head, that myosin head splits it into ADP and an organic phosphate, which you cannot see because it's hiding behind my noggin. It splits it. When it splits that, it's still bound to it, but it stretches the head of the myosin so that it's in its high energy state. And that activation is what sets it up for the pull, where it kicks off that inorganic phosphate to trigger the contraction. Still bound to ADP. My inorganic phosphate is floating off in the ether now. And it has now pulled the actin. Once it's done with that, another ATP needs to come along to kick this ADP off the myosin head to reset it. If there's no ATP around, it'll just hang out. And it will be unable to release from the actin. And this is actually what rigor mortis is. When the organism dies, mitochondria calm down, it's not getting new sources of ATP, the muscles will go through a period of constant contraction. And usually it's curling into a fetal position. And it will remain that way until things degrade a little bit further and the ADP releases from the myosin heads for different reasons. So this is actually the basis for what rigor mortis is, which is a thing that we can observe. So as long as there's enough energy in the system, there's ATP being renewed, it'll be able to kick that ADP off the myosin head and it will let go of actin and reset and stretch back out, get ready for the next cycle. And this happens constantly. It's happening every time I move my fingers, right? So yes, ATP causes the release of myosin from actin. And now I think we're ready to move on. Acetylcholine, an excitatory neurotransmitter, is the neurotransmitter released into the neuromuscular junction. This release can trigger an action potential along the length of the muscle membrane. Describe the process involved. I kind of already drew this up above, but I'm going to redo it really quickly. We have the bulb on top is our presynaptic neuron, interfaces with the neuromuscular junction. And the neuromuscular junction has these T-tubules. Okay. Muscles, if we zoom out a little bit further, are arranged in into motor units. So you'll have a nerve bundle that branches to innervate different spots of the muscle. And this is a motor unit. The contraction, I'm getting a little ahead of myself. So if an action potential is generated in the neuron, then the motor end plate of the neuron releases acetylcholine. So we get, wow, that's big. Make it tiny. We get an action potential, influx of calcium, acetylcholine binds at the neuromuscular junction. This triggers an action potential in the muscle cell, which travels down the T-tubule system. So we get a new action potential down the T-tubule, which causes its own influx of sodium, usually. But what we're trying to get to, I'm actually going to get rid of this entirely, is within this neuromuscular junction, on the muscle side, there is the sarcoplasmic reticulum. And the sarcoplasmic reticulum is chock full of calcium, just waiting to be released. So calcium is low in the rest of the sarcoplasm. It's very, very high in the sarcoplasmic reticulum. Once an action potential makes it down one of these T-tubules, and that action potential reaches the sarcoplasmic reticulum, that triggers a huge release of calcium. And this is the source of calcium that binds with the tropomyosin that we talked about a moment ago, which allows for myosin to act on actin. So that's where that calcium comes from. And then you have membrane channels that pump calcium back into the sarcoplasmic reticulum when it's done. This is important enough that I'm going to write it out again a little bit differently. So an action potential travels down the neuron, arrives at the neuromuscular, zoom out a little bit more, come on, arrives at the neuromuscular junction. Neuron releases acetylcholine, binds to muscle cell, and triggers an action potential. The action potential travels down a T tubule, causing calcium release from sarcoplasmic reticulum, which triggers contraction of the sarcomere. Okay. So now we have an action potential applied into motion in skeletal muscle. If acetylcholine always produces an excitatory response in the neuromuscular junction, how can we regulate which muscles are contracted and which are extended at any given time? For example, how can we bend the arm only partially? This is where I was kind of getting ahead of myself talking about motor units. Muscle is arranged in two motor units. And the contraction of each motor unit is controlled by separate neurons. The degree of response depends on how many motor units have been stimulated to contract. The degree of response depends on number of motor units stimulated to contract. Severe calcium deficiency can lead to a reduction in bone mass. It can also have serious effects on the functioning of the nervous system and on the action of muscles. Explain what roles calcium plays in the activity of muscles. We're not currently worried about the bones, although that is also a serious side effect of calcium deficiency. So as we were talking about up above, calcium release is absolutely necessary for a muscular contraction. Its release into the sarcoplasm again, just to review, moves tropomyosin away from actin, so myosin heads can act on actin. So with severe calcium deficiency, we would have weak muscles, so decrease muscle strength, or Yeah, decreased muscle strength due to a decrease in the strength of the contractile potential. Cool. And it is exactly 5 p.m. That is the end of our worksheet. If you have any questions, I can stick around for a couple of minutes. If not, have a fantastic week. Don't forget to submit your worksheets for that sweet extra credit, and I'll see you next week. All right. Have a great weekend, everybody.